Thank you, Nadia, for the nice overview in regenerative medicine. So now we still have some time for the discussion. I hope you have some questions ready. I would like that everybody who was presenting comes up to the stage and we can have a discussion. Okay, du bist gerade der Zweite. Okay, are there any questions? Yeah, so far.
I guess the microphone is not on, but okay.
Oh, now it's on, yeah.
I would like to address a question to Mr. Christopher Trevel with respect to the kidney model you have. Could you expand a little bit on the type of tubular cells which are present in your model? And secondly, whether you have any information on functions of this tubular system? Thank you.
Yeah, okay. The main cell type we work with in the kidney model is proximal tubular cells because they are the main target of acute kidney toxicity. So far this is an immortalized cell line, so using the TERT fragment, the cell line is called RPTEC TERT1. And as to the functionality, so what I investigate is the tightness of the layers, the polarization under static conditions and flow conditions, transport characteristics under static and flow conditions, and toxicity with well-established proximal tubal toxins like cisplatin, tobramycin, and others.
Did you also use primary cells, proximal tubular primary cells?
So far not, no. The next step or the midterm step for kidney and all other organs will be to replace it by iPS-derived models.
Mm-hmm. Are there any more questions from the audience? Please use the microphone then. Yeah.
So thank you for these interesting talks. I have a question regarding this organ on chip things. Regarding the kidney, what about this glomerular structure, which I think mostly missing in this organ on a chip system always?
Yeah, so of course, I mean, it is a very intricate structure, and so far no one has achieved in getting the slit diaphragm and the podocyte endothelial cell barrier correctly in vitro cultures. So yeah, it's the next step. So I started with this project trying to mimic the filtration barrier, but so far I cannot say I have succeeded. But of course, yes, we are aware that we need it, like a size-dependent filtration barrier, and we will try to build that in.
And maybe a second question, if I'm allowed, maybe to all who worked with this organ on a chip. So for drug testing, I assume that the main readout will be some fluid-based biomarkers within these systems. What about the morphology readout, which we usually have if we do animal testing?
Okay, basically the flow is not always the only readout. In our current system, we can sample the supernatant and do an ELISA or PCR. So that's the type of readouts we have. Also, the chip we developed, that's the long-on chip we developed, we can really open it. So you can, at the end, you can really put that on an extremely thin slide and have a look actually with a 40x, 60x like you want. And so the microscopy is not an issue. You can do immunostaining like another assay. And yes, of course, we do permeability, because that's one thing, actually, that's for the lung microvasculature. So you can really have a look whether the microvasculature is stable or not. We observed a very high significance between endothelial cells alone and endothelial cells and pericyte, for example. And also now, actually, the latest paper we just submitted between, I forgot to say, all the pericyte we use were from patients. And the next study we just submitted is between lung parasite from patient in the tumor side and just outside of the tumor, and here too we observe the permeability difference.
Are there any more questions? Okay, maybe I have one question for Simon Ströbel. I was wondering, you're using these iPS cells to produce the cardio or the heart microtissues. At what point are you differentiating them? Are they already pre-differentiated before you make the microtissues? Is this important, or could it also be possible to differentiate them once they are microtissues?
So they're actually pre-differentiated when we use them in the tissue or to produce the tissues. But it's also important to have like the connective tissue or the associated fibroblasts in the system to have this functionality.
Mm-hmm. And these fibroblasts, are these primary fibroblasts, they're human origin?
Primary fibroblasts, yes. And
also from the respective tissue, or are they from the dermis, or?
From heart, yeah, cardiac fibroblasts, yeah.
Okay. And this beating, you showed some pictures or some images about this thigh movement. So this contraction of the microtissues is synchronous. So
yes.
The whole tissue is contracting at once. There are not just parts beating.
They're all synchronized and they beat at once. Yeah, it's very interesting. Yeah.
If I may ask also in this. How big are your spheroids, and do you have a necrotic core in there? I mean, it's quite amazing that you have something that is quite homogeneous and that beats synchronously.
So they're up to about 300 micrometer. So the cardiacs, 250 to 300 micrometer, and they're functional for several weeks, as we've seen. So they continue beating. Of course, when you supply the nutrients and yeah.
Are there any more questions from the audience?
Maybe I have one question to you, Markus. So I like very much that model with the muscle fiber model, because I think that like mechanical cues are still something that is very underestimated in cell culture, and we have very positive experience of exposing cells to flow, and also things stretch and contraction are very important. So did you see an impact on your stainings of your muscle fibers of the myosin heavy chain, for example, after the contraction induction by electrical stimulus, compared to non-induced models, for example?
That was more or less our end point when we were electrically stimulating these tissues, so they were not really contracting for a long time. And we also, at some point, we also wanted to integrate mechanical stimulation to form the proper tissue during the development, but we did not do that so far. So we are planning also to electrically stimulate the tissue while it is developing at some point. But of course, it must be much stronger at the end. We are expecting much higher forces, and we don't have this nice muscle fiber formation yet. Any more questions? I would have one for Nadia. So maybe I missed it. You went quite fast through the slides. So this release of the growth factors, how is it induced? Is it you said through the cells, right? So is it enzymatic then, or?
It's cell contact dependent release. That means cell come in contact with the implant, and we have local degradation by enzymes of cells, and we have the liberation of the active molecules.
Okay, so does it degrade the polymer. So sometimes you have it encapsulated into these polymer capsules, if I'm right.
So we have the bone morphogenic protein 2, the active molecules, and excipient, as excipient we have chitosan. For example, we can use hyaluronic acid or other active excipient, non-active excipient for the nanoreservoirs formation.
Thank you. Please,
Yeah.
you use the microphone.
Yes. This one? Okay. So you showed like the improved bone regeneration when you use aggregated cells
Spheroids.
for implantation, so the double 3D implants.
Yep.
Is there an explanation for that?
So the explanation is we have a double 3D environment. That means the cells in spheroids, in microtissues, have their own extracellular matrix by collagen matrix, the natural one. And by adding these microtissues to the 3D other environment with nanofibers, they have another environment, and with this environment, we can increase the speed of bone regeneration because we have double structuration of the environment of cells. And in the double environment, 3D environment, we have active molecules. So we have nanoreservoirs of growth factors.
So it would be an additional signaling.
Yes, yeah. Okay. And that's work better than single cells cultivated on the top of the nanofibrous membranes. And that help also the migration of cells because they have their own extracellular matrix then.
Thank you.
Thank you. More questions? I would have one for Olivier. When you are commercializing this lung-on-chip device, what material is it then made of at the end? It will be injection molded, or because for now it will be this PDMS, right? So I'm just asking because of this absorption problem of the PDMS, and yeah.
Yes, at the moment, actually, the lab device, of course, is made of, everything's made of PDMS. The end device will be made partly of injection molded plastic, but the membrane will remain PDMS for the time being. Yeah, we are, of course, fully aware of the problematic there, but basically, we believe at the moment you can do a control and have a look with a control if what is the absorption at an absorption problem with a molecule and do the test next to that. I believe, I mean, I cannot tell you the secrets, but of course we also working on other type of materials and to replace PDMS, but to start with, that's going to be the material. And already today, actually, some products are on the market which are stretch the cells, and this is all done actually with silicone.
Thank you. And I think in your case, you have anyway covered all the PDMS with cells, right? If it's just on the membrane.
It's also coated actually with collagen, fibronectin, etc.
Okay. Are there any more questions from the audience? So if not, I would like to close this session, and I would like to thank all the presenters for their nice presentations and fruitful discussions, and I would like to thank you again for your attention. Thank you.